Welcome back, champions. Let's get straight into it. So this first video is going to be about enzymes, which is covered in both Unit 3 Area of Study 1 and Unit 3 Area of Study 2 for VCE Biology 3-4. So this video is going to be split into two parts. Firstly, I'm going to be discussing enzymes and a quick introduction to them. And then I'll be moving on to factors influencing their activity. So let's get straight into it. So introducing enzymes. So first, you have to know that chemical reactions have to occur in order for life to be sustained, and that they only occur when there is sufficient activation energy to break the bonds in order to start the reaction. So here is where we're going to talk about anabolic and catabolic reactions. So first of all, anabolic reactions. So you can think about these as build-up or synthesis reactions, wherein two or more smaller reactants combine to make a larger one. And you can think of energy being stored. And as a result, the energy of the products is going to be higher than the energy of the reactants. So here we can see an energy profile diagram of an anabolic reaction. So the EA is the activation energy, and the delta G is the enthalpy change. So that's a difference in the energy between the products and the reactants. So as you can see here, the energy of the products is higher than the energy of the reactants. Okay, the next one. Catabolic. Okay, so I think of catabolic as breakdown reactions. So you can think about how one large molecule gets broken down into several smaller molecules. So in this case, energy would be released. And as a result, the energy of the products is going to be less than the energy of the reactants. So as you can see here, the energy of the products is higher, and this eventually lowers during the progression of the reaction to the products which have a low energy. Okay, now let's actually go into what enzymes are and what their function is. So this is very specific terminology you need in order to get all your marks, and trust me, you really need these. Okay, so they are biological catalysts, meaning they speed up the rate of reaction by reducing the activation energy required for the reaction to occur, and are also recycled, which means they aren't used up in the reaction. And they're said to be biological because they help catalyze biological pathways and reactions. So here are some additional notes that contain some additional information that is important to know and may be important in answering questions. So most cellular reactions require enzymes to occur, and without them, the reaction wouldn't occur at a fast enough rate to sustain life, meaning the organisms would die, and that is obviously not good. So we can look at it here. So as you can see, the activation energy without an enzyme is higher than the activation energy with an enzyme. And that is the same for both catabolic and anabolic reactions. So another thing is to state is that they are specific. So that means they only catalyze one type of chemical reaction. So this is due to the shape of the active site only being complementary to the tertiary structure, which is to say the 3D shape of the specific reactants that they catalyze. So how do enzymes actually catalyze these chemical reactions? Well, here's a little pathway we can look at. So first of all, this is the substrate, and this is the active site of the enzyme. And what happens is the substrate enters the active site of the enzyme. And as we see here, the enzyme might slightly change shape as a substrate binds, and this is referred to as the induced fit hypothesis. And then the reaction occurs, as can be seen by changing the color of the reactants into the products. And after the reaction has gone to completion, then the enzyme releases the products and can be reused for future reactions. So like I said, it's important to remember that enzymes lower activation energy. Okay, so let's look at them specifically for anabolic reactions then. So for the anabolic reactions, as you can see here, they bring the reactants closer to the state in which they need to be in order to react. And as a result of that, they reduce the activation energy required for the reaction to occur, which is why the rate of reaction increases, since more of the reactant molecules have sufficient energy during collision for the reaction to occur. And that increases rate of reaction. Now, catabolic reaction is obviously opposite of this, as a catabolic reaction, as was covered earlier, is a breakdown reaction. So this places stress on the bonds of the reactants, and as a result reduces the activation energy required for the reaction to occur, as there is less activation energy required because there's already stress being forced on the bonds of the reactants, allowing them to be more easily converted to the products. Again, meaning that more reactants have the sufficient activation energy for the reaction to occur, and therefore increase rate of reaction. So here are some additional features of enzymes. So first of all, they are reusable, which means they can be used up for multiple reactions and are not consumed in the reactions. They are specific due to the active site only being complementary to a few or a single reactant. They're reversible, mostly, not all though. And they increase the rate of and not the yield of. And this is a very common misconception. They only increase the rate of reaction that a reaction occurs at, not the amount of products produced. And additionally, it's important to remember that they are proteins. And they are also a subset of catalysts, as they are organic. But catalysts can also be inorganic. And they act on entire biochemical pathways, which will be discussed in the next part of this lesson. And they typically end in ASE, and this is quite common, although not all enzymes will follow this, but it is important when trying to identify them. And they're typically written above the arrow. Okay, let's go to the next one. Factors influencing the activity of enzymes. So first of all, let's go through some recall practice to make sure you understood everything that we learned in the previous part of this lesson. So I'll give you 10 seconds. Try to fill out all these spaces. All right, so you can pause the video if you need more time. So let's quickly go to the answer. So this would be the substrate, as this is the reactant that is being acted on by the enzyme. And the shape of the substrate is complementary to this part of the enzyme, which means it must be the active site. And as a result, this is the enzyme. And once a substrate is bound to the enzyme, it's referred to as an enzyme substrate complex. And after the reaction has occurred, then the enzyme releases the products, as can be seen here, and can be used to facilitate more reactions. Okay, now let's go through some factors that impact the functioning of enzymes. Okay, first of all, let's go through temperature. So, as temperature increases, the particles have more kinetic energy, which means they move around quicker. As a result, enzyme activity increases the rate of collisions between reactants and the active site of the enzyme at a sufficient energy for the reaction to occur, meaning that the activation energy is surpassed. And as a result, the reaction can occur, meaning there are going to be more successful reactions and more successful collisions. So, as we can see here, this is the optimum temperature as seen by the blue dotted line. So we can see that the enzyme activity is highest here. So the optimum temperature is the temperature at which the enzyme is most active. And it is important to note that after the optimum temperature has been surpassed, the enzyme activity reduces, as can be seen here, as a result of the enzyme's active site becoming denatured due to the tertiary structure of the enzyme changing. And this means that the substrate is no longer complementary to the enzyme active site, which means that it can no longer bind and form a substrate enzyme complex, and as a result won't be able to catalyze the reaction. Okay, so next factor is pH. Okay, first of all, the rate of reaction is going to be greatest at the optimal pH, as can be seen here. So similar to optimum temperature, this is the pH at which the rate of the enzyme catalyzed reaction is the greatest. So as you can see here, for example, pepsin, the rate of reaction at which the optimal pHs would be around 1.5 to 2. And as we can see here, the rate of reaction abruptly drops off as pHs increase or decrease from this optimal range. And similar to temperature, as a result of the change in the pH out of the optimum range, it results in the enzyme's shape being denatured, which means the tertiary structure of the enzyme changes, and as a result, the active site is no longer complementary to the substrate, which means that a substrate enzyme complex can no longer be formed, and as a result, the enzyme can no longer catalyze the reaction. And this would mean that fundamentally, the rate of reaction will be reduced as a result, as we can see here. So some additional notes. Some enzymes tend to work in a wider range of pH conditions, for example, alkaline phosphate, whereas other enzymes are more sensitive to pH changes and may only work in a more narrow range, such as pepsin. And this change in pH has the potential to affect the interactions between amino acids in the peptide chain that forms the enzyme, which impacts both the primary and all the above levels of structure, which can result in the enzyme becoming infunctional. And like I said before, this changes the shape of the active site. All right, next factor is going to be substrate concentration. So, as the concentration of substrate increases, the rate of reaction proportionally increases as well, since the number of successful collisions between the enzyme's active site and substrate increases to a point. So, at this point and past this point, the rate of reaction plateaus, which means it flattens out, so it doesn't increase further. So, this occurs at what we call is the saturation point, which is the point at which all the active sites of the enzymes are occupied by the substrate. So, even if we were to theoretically increase the substrate concentration, the rate of reaction will no longer increase because there are no longer any more active sites of the enzymes available to be able to be occupied by the substrate to allow to facilitate the reaction, which means that the rate of reaction will remain constant after this point. So, enzyme concentration. So, similar to the effect of increasing substrate concentration, as a result of increasing enzyme concentration, the rate of reaction increases proportionally as a result of the increase in the frequency of successful collisions between the enzyme's active site and the substrate, up to a point like before. So, the first reason is that the substrate concentration might be a limiting factor, which is a factor that prevents the rate of reaction from occurring at its optimal, which means that it can no longer happen at the highest rate of reaction. Or the second factor or reason might be that the end product of this enzyme catalyzed reaction has begun to regulate the activity of enzyme catalyzed reactions near the beginning of the biochemical pathway. So, as we can see here, this product, which is product 3, then goes back to these enzymes that facilitates the first step in the biochemical pathway and changes the shape of the active site, which means that the substrate can no longer bind and the reaction is no longer catalyzed, and as a result, the rate of reaction reduces. So, enzyme inhibitors. So, these are molecules that bind to and prevent/reduce the functioning of an enzyme. So now there's two types. We have a competitive and a non-competitive inhibitor. So the competitive inhibitor, and this is a key word, competes with the substrate in binding to the active site due to having a similar shape to the substrate. And when the competitive inhibitor is bound to the active site, it means that the enzyme is occupied and the substrate can no longer bond, which means that the enzyme substrate complex can no longer be produced and the reaction can no longer be catalyzed. Now let's talk about reversible inhibition. So this is when the inhibitor can form a weak bond with the enzyme's active site. However, as a result of forming a weak bond, This can be overcome by increasing substrate concentration, which increases the chances that the substrate will bind to the active site as the enzyme, as opposed to the inhibitor, allowing the reaction to increase to its original rate, as we can see here. So this is without any inhibitor. So it increases and eventually plateaus as a result of reaching the saturation point. But with a competitive inhibitor, it is initially inhibited, but as a result of increasing the substrate concentration, this overcomes the effect of the competitive inhibitor, allowing the rate of reaction to increase to rates as previously without the inhibitor. So now let's move on to non-competitive inhibitors. So as opposed to competitive inhibitors, which compete with binding to the active site, which is where the substrate binds to, the non-competitive inhibitors instead bind to another site on the enzyme known as the allosteric site. And as a result of being bound to this allosteric site, it changes the shape of the active site, which means that the shape of the substrate is no longer complementary to the shape of the active site, and substrate complex can no longer be produced, and the reaction can no longer be catalyzed. So now let's talk about irreversible inhibitors. So these are inhibitors that form a strong bond with the enzyme's active site. So as opposed to reversible inhibitors, these cannot be overcome by increasing substrate concentration. So this means that the enzyme is prevented from reaction indefinitely. So here's another graph that we can use to visualize this. So this is a normal enzyme without any inhibitor. So like we discussed, it increases as substrate concentration increases, but eventually reaches a saturation point as which it plateaus. But in the case of a non-competitive inhibitor, it plateaus much earlier as a result of this irreversible inhibitor forming a strong covalent bond with the active site that prevents the substrate from binding, meaning that it plateaus immaturely. Okay. So we kind of delved into this previously when talking about factors that influence enzyme concentration and that cause the rate of reaction to plateau as a result of this. But now let's go into it a bit further. So some enzymes have more than one binding site. So for example, one for the substrate and another one from the product of the biochemical pathway. So the end product binds to the second binding site of the enzyme, which acts in a similar way to an allosteric inhibitor, or to say a non-competitive inhibitor, where it changes the shape of the active site and therefore prevents a substrate from binding, and as a result, prevents the enzyme substrate complex from being formed, preventing the reaction from being catalyzed. So therefore, this regulates the amount of product produced, and this ensures that extra energy isn't used when it is not required, and this increases the chance of survival of the cell. So now let's go into cofactors and coenzymes. So cofactors, these are organic or inorganic molecules that assist in enzyme functioning. So as we can see here, in the inactive enzyme, the cofactor is not bound to the active site. But then, after the cofactor bounds to the active site, it is referred to as an active enzyme, as it can now allow the reaction to occur since the substrate can bind, as is illustrated. So now let's talk about the importance of these. So without the cycling of cofactors, many biochemical processes wouldn't occur. And don't worry if you haven't covered this yet. So this refers to cellular respiration and photosynthesis. And that will be covered in a later video. And also ensures, similarly to an enzyme, that the rate of the biochemical reaction in the pathways occur at a fast enough rate to sustain life. Now let's talk about coenzymes. So coenzymes are a subset of cofactors that are inorganic, non-protein molecules that aid in enzyme catalyzing reactions. So this is an illustration of how they work. So first of all, as we can see here, this is the inactive enzyme. And after the coenzyme binds, it changes the shape of the active site, which therefore allows the substrate to bind. And the coenzyme may also donate some chemical groups which provide energy to ensure there's sufficient energy that surpasses the activation energy for the reaction to occur. And then after the reaction occurs, Since the catalyst is not used up, it reverts to original shape and can be used to catalyze future reactions. And as for the products, they just go away. And the structure of the coenzyme is changed and can only be converted back into its loaded form if it is supplied chemical energy to allow these groups to be reformed. So this is what we're referring to when we're going to talk about loaded and unloaded coenzymes and cofactors. So when I say loaded, I mean that it can release stored chemical energy by donating chemical groups. For example, this coenzyme is loaded since it can release chemical energy by donating chemical groups to allow for this reaction to occur, to allow for the activation energy to be surpassed. Whereas when I say unloaded, is a loaded enzyme that has already released energy by donating chemical groups. As you can see here, this coenzyme is unloaded, basically can no longer facilitate more reactions and must be cycled back into its loaded form. And this is what we're talking about when we refer to the cycling of cofactors, and that being essential to biochemical pathways. Alright, so thank you so much for tuning into me this video. I hope to see you again for future videos, and as always, feel free to post your questions in the comment section below. Thank you.